So we are talking about, because we are saying that neural tube formation happens because you have the change in the shape of those cells, right? That cause folding and ultimately formation of that tube. So how that happen? So we are saying that BMP signaling induces the both the hinge plate cells, which are like medial hinge plate, MHP, and dorsolateral hinge plate cells. So what we know that what happened? We know that when the cells are right here, so these were the cells of the neural plate. This is neural plate. And we say that neural plate come in contact with the notochord. When it comes with contact with notochord, they shorten and they ultimately change the shape. So the shape become like this, right? So the cells which were like this, they were like this, they changed to this shape. So if you look here, what is happening here, if this is the apical surface, this is the apical surface, and this is the basal surface. So apical surface has remained, if I draw it bigger, apical surface, this is the basal surface. So if you look at the apical surface, this apical surface has changed the shape, right? The basal is still the same way as it was. So what is happening there? We are saying that BMP signaling play a critical role here. So on the apical surface, so this is the apical surface, this is the basal surface. On the apical surface, what you have here, you have right here on this side, these are the BMP receptors. So BMP receptors are there. So when you have BMP receptors, what happens? That these BMP receptors, they receive BMP. So this is BMP. So when they receive BMP, these BMP receptors are activated. So that means receptor activated. So BMP join to BMP receptor, that means activated BMP receptor. Okay. Then what that does? When the BMP receptors are activated, they recruit SMAD. So activated BMP receptor, oh, I'm sorry, should have kept it now. So, okay. So BMP receptors, they basically activate SMAD. So what happened? So this is the SMAD. So BMP activator, they are activating the SMAD, so SMAD is there. So once the SMAD is activated there, so SMAD does two things. Right here is the nucleus. So SMAD activation means the SMAD activated SMAD. I would say SMAD activated. So activated SMAD is there. So some of the activated SMAD from here goes to the nucleus. So this is activated SMAD goes to the nucleus. Okay. And then what happened? That this activation of SMAD also stabilize right here on these sides. on both the sides here, these are the polarity complex is stabilized. And what is in the polarity complex? You have power 3, you have power 6, and atypical PKC. So that is stabilized here. Okay. So when that's stabilized, what that does, that leads to stabilization that leads to equal so idea here is what you want here is you want this cell to stay like this. It means there should be something which can keep these cells like this. Okay, so we call them basal defining complex, also called LGL. So when this happens, throughout the cell, these basal defining complexes, those are kind of distributed throughout the cell like this. So this is LGL, basal defining complex, also called LGL. It's called lethal larva, lethal gene larva, something like lethal giant larva actually. This is from lethal, L-E-T-H-A-L, lethal giant larva. So that's how they discover this, like those larvae which were having mutation in this complex, they were all dead. So they call them lethal giant larva chain in the beginning. So what we call them basal defining complex. So let me go back, what happens. So we are saying in the cells of these cells which are here, which are making neural plate, these cells show at the basal as well as the apical as well as the basal epithelia, they have the same length, right? And they have perfect rectangular type of cells. We are saying that is maintained by the BMP signaling. We are saying on the apical side of these cells, they have BMP receptors. When those BMP receptors are bound to BMP, what that does, that activates SMAD, and activation of SMAD recruit polarity complex at the basal side, and that polarity complex is composed of power 3, power 6, and atypical PKC. And when that happens, some of this SMAD, which is phosphorylated now, goes to the nucleus, and that induces changes that cause equal distribution of basal defining complex, also called lethal giant larva, in the cell, and the cell maintain its symmetrical position. That is okay so far? Any doubt here? Yes or no?
So the SMAD and the nucleus causes the LGL.
Yeah.
Okay.
Okay, then what happens? So I'm just erasing this thing. So what is happening there? Right here is your nodopode, right? So you will say from where the BMP is coming. So this is the neural plate, the cell surrounding here, which were on this side, what they were doing. So this was your neurocast, this was your epidermis cell, right? And remember, they are epidermis because they are making a lot of BMP. So it means cells here, on this side, they are secreting a lot of BMP. So a lot of BMP is there, right? So what happens? notochord is the place we said that's going to secrete BMP inhibitors. Okay? So notochord, which is here, that start making, that we know that it makes BMP inhibitor. That's why we said like we have head and tail thing happens. So notochord here is making BMP. Oh, sorry, not BMP, Nogin, for example. So these are the BMP inhibitors. So these cells are right above it. Okay? So it's sending BMP inhibitors here, BMP inhibitors are there, BMP inhibitors are there. Okay? So BMP inhibitors are being secreted there. What that does to these cells, which are cells here, so then, if the BMP inhibitor is there, what is going to happen? So this BMP molecule, this is BMP molecule, and then BMP inhibitor, for example, say here is Nogin. So if that is there, then what is going to happen? So for example, this is the receptor right here. Here, no BMP. If there is no BMP, that means therefore no activation of SMAD. So there is no SMAD here. If there is no SMAD there, then what is going to happen? That polarity complex cannot be stabilized at the apical end. Therefore, no stabilization of polarity complex on the apical surface. So no stabilization of that. If there is no stabilization of that, then there is therefore no distribution, no even distribution of LGL. So what happens? What that does, that leads to a stage like this, because there is no LGL here. All the LGL, so the polarity proteins are here, so this is the polarity protein. They are like randomly now distributed. They are down here where they should be, right? So these are the, so this is your polarity complex, polarity protein complex. And then the LGL is all here. Basal defining complex is all there. And when that happened, that caused these cells to change the shape like this. LGL, this is your polarity complex. And that leads to the change in the shape. So what is happening here? So we are saying notochord is directly, because these cells are in contact with the notochord, right? So these cells are in contact with the notochord, and then notochord is kind of like telling these cells, hey, that I'm going to take away the BMP. If the BMP is not there, BMP receptors are not activated, BMP receptors are not activated, this polarity protein complex is not stabilized at the apical surface. That means no SMAD activation, no SMAD going to nucleus. That means this basal defining complex, LGL, is not evenly distributed. And that all LGL comes to the basal surface, and that causes this side to remain stretched and this side constrict, and that causes the change in the shape of this cell. Making sense? Right? So talk about this for a minute, then we'll go further.
MHP and DLHP. It should be a dorsal
Okay, any question here? Any confusion? No? Any confusion here? Any confusion? Okay. So then, actually we did a lot of work on this polarity complex. So we discovered how you have a neuron with only one axon, a neuron can have two axons, neuron can have three. If you go to PubMed, you put my last name and first initial and just put part 3 there, you'll immediately go to that paper where we describe about how these proteins are critical for making one neuron with one axon, two axon, three axon, all those things. So then, here is then situation occurs. So we're still on the same thing, right? So what is happening there, we are saying, so this is your epidermis, and then we are saying that these cells are like this, right? So these cells are the cells which are basically making your MHP, middle hinge point, right? And right here is your notochord. Now here is one more thing happening. The cells here, if you look here, so let me draw these cells. So here was the deal. These cells change the shape, right? Then we said none of these cells change the shape. If these change the shape, if these cells also change the shape, then it will be like kind of crumbled like this, right? These cells, but then we said that cell right here at the dorsal hinge point, they also change the shape, right? Only then the closure takes place. So what is happening there? So just pay attention what is happening here. Notochord, so now these cells which are here, all these cells which are right there, they are making a lot of BMP. Right? Because these are epidermis cells, they are making a lot of BMP there. Now, this is epidermis. They are making a lot of BMP. They are secreting BMP. So it means everywhere here you should have BMP, right? So that's why these are in direct contact. They are not, but Nogin is also there, right? Because these are also secreting Nogin. But there is like too much BMP there, BMP coming there. That's kind of like, this is now inside, this is outside. So BMP is kind of like affecting those cells. But they are also, this notochord is also secreting Nogin there. But then notochord is also secreting one thing called sonic hedgehog. Now you will say, there are three things I'm talking here. I'm talking BMP, then I'm saying inhibitor of BMP, right? And I'm saying a lot of BMP is there, but Nogin is also there. So Nogin should inhibit BMP, right? If Nogin should inhibit BMP, like it did here, very close, then all those cells, they should change the shape. But this does not happen. Because this sonic hedgehog, which this is secreting here, now these are in direct contact, they're directly getting the Nogin. But the sonic hedgehog which is made here, that sonic hedgehog is actually inhibiting the Nogin. So sonic hedgehog inhibits Nogin. Now, these cells are very near to notochord. Notochord is making a lot of sonic hedgehog, so here everywhere Nogin is inhibited, so BMP is showing effect. When it's going far away, the sonic hedgehog cannot reach in this area. It's far away. But Nogin can reach, right? So right here on this side, because sonic hedgehog cannot reach there at this area, so BMP is also inhibited at this point. So here also BMP is inhibited. And therefore you have dorsal hinge cells are formed. Making sense? So that's why there are two places where these hinges are formed. And that hinge formation is controlled by the notochord. Two factors, it's sending inhibitors of BMP and also sending inhibitors of Nogin. Making sense? Yes or no? Okay, then talk to each other for a second, then we'll go further to the next topic. Make sense?
Yes.
Any doubt?
I don't believe so.
Any doubt here? You're okay? You're fine? Okay, good. Now, we are at the next topic, and we are going to call it events of neural tube closure.
This is.
Events of neural tube closure.
This is subtopic.
So this
Say,
we
what,
just
what is that?
We just finished topic 8.
No. We are at the subtopics. So I'm giving the heading of what we are covering. Like I just provided the previous
Okay.
heading, you know?
Yeah.
The subtopics of the topic.
Right.
Yes, we're still on the nervous system formation. Now, think about the embryo was like this in the beginning, right? And then we are saying it changed like this. right? So what is happening in the, so we are all amniote, right? So in amniotes, body axis elongates, and it elongates from interior to posterior. So it means that the neural tube formation, like that thing starts, right? So that thing, for example, starts here and move from here to here. Making sense? So ultimately we are saying a tube is formed. And that tube that is formed, that tube doesn't form evenly at the same time. Let me show you one thing. You understand that here. So this is the chick embryo showing the 24 hours after fertilization. So look here. Here you can see the neural fold, neural groove. You can see that neural tube formation has started, right? Here, very close, right? There's slight difference. And here, just the first hinge has formed, right? Here, still the gastrulation is taking place. Because that is elongating, so usually the anterior side is more progressive or ahead of the posterior side. So that means that neural tube formation does not start at the same time throughout the axis. If it does not start at the same time, that means the tube will also not close at the same time. Making sense? So what is going to happen? Ultimately, you are going to form a tube like this. So a tube is formed like this. And this tube that is formed, this is your neural tube. Now, this tube naturally will have two pores, two holes there, right? One hole here on the tube, and that is called anterior neuropore, and posterior neuropore. But these are four here and here of that tube, right? But then we are saying that this tube also does not close at the same time. It means some places it will be lagging behind, right? So it means there are multiple places where this tube actually closes. So let's talk about what are those places where the tube is going to close. So if I'm drawing this human brain, okay, so let me draw this tube like this. So that tube should be like this. Okay? So there are at least 5 different closure points at which this neural tube closes. Okay? So then there is this side right here where the neural tube closes. This is called closure site 1. And this site is where this is junction of hindbrain to the spinal cord. So this is the closure site 1. That's the first place where this closure takes place. Then we have this closure site right here. This closure site is called closure site 2. Okay, and the closure site 2 is where forebrain and midbrain this is the place where forebrain and the midbrain are there. That's the second closure point. Now then you have this point, this tube should be closed from here also. This tube is closure, this is called closure site number 3. And this is what we call it, this is the rostral part of the forebrain, okay? So we say rostral forebrain is closed there. Then you have the closure site 4. This is the closure site 4. And this closure site 4 is the junction of hindbrain, junction of spinal cord and midbrain. That's closure site number 4. And actually this is even before the spinal cord, so this area before the spinal cord we call them medulla oblongata. So usually we call that area as hindbrain or brainstem. So I will write here actually brainstem instead of, so brainstem, that will be more correct, and midbrain. Brainstem and midbrain. Now, there can be, these are, we are saying, a very easy thing to say, okay, this is what is happening. Now, neural tube closure is critical. Every 1 in 1,000 live births, 1 in 1,000 live births, you can have neural tube closure defects. So if neural tube doesn't close properly, then you will have neural close defect there. Now, most important defect occurs at the position number 1, site number 1. And that is very common, and that leads to spina bifida. That's a very common place where you have all the neural tube closure effect take place. Okay, and then, oh, I forgot to give you one more thing. That is here, the closure tube. This should also be closed, right? Because this is on the end. So this is the closure site 5. This is the closure site 5, which is posterior neuropore closure. So, most common is spina bifida, and then you can have problems in closure site number 2 and closure site number 3. These sites may not close. When these sites do not close, that is lethal. So 2 and 3 closure defect is lethal. That causes a condition what we call anencephaly. Anencephaly. What happens in that condition? That the brain or the developing nervous system remains in contact with the amniotic fluid, and ultimately the nervous tissue it degenerates, and there is no skull formation in that case, and that's a devastating stage. And here is actually a picture there, I believe. So this is the patient, how it looked with anencephaly. Actually, there is no brain formation there because it was open and that was in contact with amniotic fluid, no skull development, all the brain. And this kid died in 2.5-year-old kid that died. So then there are multiple genes which are responsible for this closure. right? And those genes are right here. Genes responsible for neural tube closure. Those are sonic hedgehog, that's one gene. Grainy head is the another gene, and then you have the PAX3. These are important genes which are responsible for the closure of this neural tube. Now, interestingly, I am telling you there are many effects which happen there, right? But what is happening there? There is no loss of mutation. There is no loss of function mutation. There is no loss of function mutation for any of those genes. Okay? So if you say, okay, if there is no loss of function for these genes, then why these diseases happen? This is an important thing to understand. So if there is, so that means the patients are having there are patients, right, which are showing these defects. And you're saying that these genes play important role, critical role. And then we are saying there is no mutation as such which cause the loss of function, and these patients don't show any mutation. Then they say, what is the problem there? Then they say the dietary factor. The dietary factors are important. Now, in those dietary factors, number one is cholesterol. Cholesterol is bad for you, that's what you're hearing from time immemorials, right? People say don't eat cholesterol, blah, blah, blah. But cholesterol is important factor in sonic hedgehog signaling. So it's a very important factor in the signaling pathway. So cholesterol is a very important factor in the sonic hedgehog signaling. So if you take out cholesterol totally, you're going to mess up with the sonic hedgehog signaling. Right? That's the first thing. And then folate. One of the form of vitamin B. Right? Vitamin B, folate. Folate deficiency is the main cause for the spina bifida. So during pregnancy, doctor says, okay, take multivitamin, especially folate. That's the reason. And in the countries which are not developed, they actually provide folate to go to places to provide folate. So what folate does? Folate is an important factor in the cell division. So cell division because when they are dividing these cells, they need to close, right? So cell division somehow is regulated by the folate. So you need folate there. And then there is another thing, valproic acid. You know what is valproic acid? So valproic acid is very common antidepressant. So these are kind of like tricky things. They say that the pregnant ladies, they undergo depression, they feel kind of manic syndrome and all those things. So remember I was talking about the polarity. So usually people give lithium chloride, one of the things for manic syndrome. If you read that paper which just show you there, actually we show that lithium inhibits the polarity protein complex formation, and that can cause so many neurological disorders there. Same way antidepressant drug, valproic acid, that can cause spina bifida. And then people say, hey, you don't eat fat, you avoid cholesterol, blah, blah, blah. That is also a risk factor. So there are multiple factors by which you can have the neural tube defects, and those are because of the closure. The closure does not occur. Spina bifida is not lethal, but all the other, anywhere it happens, those are lethal. Making sense? Yes.
So the valproic acid is bad?
Yes. Yeah.
And it's an antidepressant?
That's what antidepressant is, valproic acid, yeah. Okay, so far,
What did you say lithium inhibits?
lithium inhibits GSK3, glycogen synthase kinase. Remember when we talked about that's important beta-catenin signal and all those things. But GSK3 is also important for the polarity protein complex stabilization. So PAR3, PAR6 stabilization is inhibited by lithium. And that is because the GSK is in the same complex. So if you read that paper, you'll know a little bit more about that. Any other question? Any doubt here? What we just talked? Talk to each other for a second and see if there's any doubt.
I think it's a combination. Like not one that you just take. I mean, sorry, not one that like you just have.
Any doubt? You're okay here? Okay, good. Okay, now this was the primary neurulation, which we said happens till the end of the spinal cord. Then we said that if there is tail, then there should be a secondary neurulation there, right? And that happens in the mammals. There are many mammals with a tail, right? So we are now going to talk about secondary neurulation. Okay. So what happened? This is happening right at the tail. So you're having number one, it's a multi-step process. Number one is the production of mesenchyme cells. From ectoderm and endoderm. So this is the ectoderm area, and this is the endoderm. Remember, this is happening at the blastopore lip after all the gastrulation has taken place. They're kind of like cells which are stuck there in the end, right? So what happened? So these are cells here. So these cells, they basically start forming, they start dividing and start bringing cells here. And these cells are becoming mesenchyme cells. So ectoderm and mesoderm, ectoderm and endoderm, they both contribute cell, and these cells become mesoderm cell. Okay? Then what happened? Mesenchyme cell, they condense. So they condense and they form a structure called medullary cord. So they are basically condensing like this. They condense and form a structure called medullary cord. Now what happened? Central portion of medullary cord undergo cavitation. So this medullary cord was there, for example, in the center, then a cavity start formation takes place. Okay? And then once that is there, right here was your neural tube which was there. Now a cavity is formed like this, it essentially joined like that. Making sense? So this is a very easy step. So essentially ectoderm and endoderm, they contribute cells and they become mesenchyme cells. Those mesenchyme cells, they condensate and they form a condensed structure, what you call medullary cord. And the medullary cord in the center, the cavitation starts. And then this is the secondary neural tube formed by the secondary nervation in the tail area, right? And that fuses with the primary tube. Making sense? Now, we said that SOX2, SOX1, 2, 3, we said these three genes are critical for formation of neural plate, right? That's what we said. Because you have BMP and uterus there, then these are there. Wherever they are going to form the neural plate. So these cells are critical for neural plate formation. Now, when the neural plate takes place, so this is for example neural plate. This neural plate is going to give you the neural tube, right? So if the cells are responsible for formation of neural plate, therefore they are also responsible for formation of neural tube, right? Now, if you remember, this is the notochord. This is the area above this. Now think about critically that noggin is here also, noggin is here also, noggin, cord in follistatin and sending three things, right? So they are throughout. So you can have here also, you can have here also, it might spread out. So wherever it is, you will have SOX1, SOX2, SOX3, so you have to limit that spread. So then there is a protein called TBX6. That TBX6 actually inhibit, that limits the expression of SOX genes. Okay, so normally you should have one neural tube. So if you knock out TBX6, so here usually you have one neural tube in the embryo like this, but if you knock out the TBX6, what you will get, you will actually get multiple neural tubes. Making sense? So that means that every gene or every protein is somehow controlled by other protein. That's what we have seen so far, right from the beginning. We're talking about beta-catenin, GSK3 beta, all those things, VEST, everything is controlled. They're very much specified to area where they should be. If that balance is disturbed, you get so many problems, right? So now the neural tube is formed, right? Now we want to see, we said tube, we are not tube. The brain is tube, of course, but it's not tube. It has a very special structure, right? So you have forebrain, hindbrain, how that happens. That's our last thing. This is clear, any doubt here? So you can talk to each other for a minute, then we can.
Yeah, I wrote the one up here, the LGL. I don't know what I wrote. It was not LGL. Until he said like lethal diamorph, I was like, oh. Oops.
Any doubt? Any doubt there? Okay, now you have this tube being formed. right? This tube is closed here. Remember, this tube was like this, right? Now this tube is closed. So these are all your cells here. And these cells, they will not remain a single cell here, they will divide, give you multiple cells. Now what happened, once this so this is neural tube. And this tube is going from anterior to posterior, right? So what is happening, so we are talking about formation of, I'm not saying brain, but brain-like structures. That's what we want to talk about, how they're formed. And we essentially say primary and secondary vesicle formation. Primary and secondary vesicle formation. So what happened, this neural tube, it start forming vesicle-like structure like this. It will form a structure like this. And in the end, it will be just like this. It forms a structure like this. Okay. So this first vesicle which is formed here, this is called prosencephalon. This is called prosencephalon. And this cavity is going to give you the forebrain. Okay. Then the second, which is here, this will be called mesencephalon, and this is going to give you midbrain. And this third cavity right here is going to called rhombencephalon, and this is going to give you hindbrain. And rest right here, this tube will remain there. This tube is going to give you the spinal cord. Spinal cord is still a tube. Remember, spinal cord is surrounded by the vertebrae. It's still a tube-like structure, right? Now, what happened, the next step takes place, that this prosencephalon is actually divides into two vesicles. A structure like this, and a structure like this. So this is giving you these two structures. Okay. So this is called, so this structure is called telencephalon, and this is called diencephalon. Okay. So prosencephalon has given you telencephalon and diencephalon. So this is going to give you, telencephalon is going to give you olfactory lobe, which are for your smell. Then it's going to give you hippocampus, memory function, and cerebrum. Cerebrum is the two cerebral hemispheres which are there. It's going to give you that. Then diencephalon is going to give you optic chiasma, where the optic nerve is going to form. That is going to form. Then it's going to give you thalamus and hypothalamus. Okay. Then the mesencephalon, which is this area, it does not, it stays as a single vesicle. So this is still the mesencephalon. This is still the mesencephalon. And mesencephalon is essentially a center for your emotions and motor activity that is being controlled from that place. And also you can sense temperature, emotion, temperature, a sensation. Essentially this is your sensory area. Okay. Then this rhombencephalon, which was here, it also divides into two areas. So rhombencephalon also give two further vesicles, and that is called metencephalon. Metencephalon is going to give you cerebellum and pons. Those two structures are going to give you that thing. And then you have the myelencephalon. Myelencephalon is going to give you brainstem. Okay, and brainstem is usually medulla oblongata, that's what we call them, medulla oblongata. And after that, this tube, it remains as the same. This is your spinal cord. So if I have to draw the brain, for example, let me draw it like this. I'm drawing the mouse brain actually. So this is the structure of the mouse brain. Okay. So these are olfactory bulb. They are for smell. They are very well developed in the mouse. Then these two are cerebral. This is actually the cerebrum, left cerebral hemisphere, right cerebral hemisphere. And under these areas, you have hippocampus and all those things. You cut open and see the anatomy. And if you really want to see the anatomy, next semester I can show you when we have the mice in the lab. So we can open the brain and we can see what actually it looks like. And this area right here, this is your cerebellum. And beneath the cerebellum, there is pons, under the cerebellum. So this is cerebellum is sitting on the pons, right? And then this is your brainstem, or medulla oblongata, and this is your spinal cord. So what we are saying? We are saying that brain formation takes place when this neural tube vesiculate. Initially, this neural tube vesiculate and give three primary vesicles. Three primary vesicles which are formed. Prosencephalon, which is going to ultimately give you forebrain. You have mesencephalon going to give you midbrain, and you have rhombencephalon going to give you the hindbrain. And we said that this process proceeds further, and secondary vesiculation takes place. So prosencephalon give you two vesicles, telencephalon and diencephalon. Telencephalon is going to give you olfactory lobe, hippocampus, and cerebrum. So all this area is coming from there. And then we are saying that diencephalon is going to give you optic chiasma, or your optic nerve is going to go there. So that means the vision center, giving you thalamus and hypothalamus. Then the mesencephalon cavity or vesicle does not divide; it stays at the same one vesicle. And mesencephalon is a center of emotion, motor activity, temperature sensing, all those things take place. Then rhombencephalon also vesiculate in two small vesicles. Metencephalon is going to give you cerebellum and pons. And then we are saying the other vesicle is myelencephalon, which is also coming from the rhombencephalon, is going to give you medulla or the brainstem. And further along, this neural tube below this is already in that spinal cord formation. It remains as a tube. Now, here is important thing, and I will start that Monday with that. That think about in our body. Brain is right here in the skull. Spinal cord is in the vertebral column. All the cells which are making neuron, they are staying here. So all those are going to give you billions of cells, right? So all the cells are here. Think about the human body. If this is the human body, let me just draw it here. If this is the human body, and rest body is here, you know, arm, legs, all those things. So brain is here, so all the cells are here. All the cells are here, right? So, but you are moving your arm, all the muscles everywhere there. There is no cell. What is there is just the axon coming from here, axon coming from here, axon coming from here. So no cell from our brain actually directly talk to the body. Brain, so all the message, for example, someone touch you, from here through these axodendric thing go here, these are sensory, goes to spinal cord. Spinal cord, if say, okay, it's an immediate thing, do it immediately. It sends signal to these muscles and you move it. Or if it's a long time you're thinking, should I go out or should I not go out? Then you send the signal here, it send it back, say, okay, let's move. So brain always talking to spinal cord. Spinal cord is talking to rest of the body after receiving these signals. So it means here you have only wires. There's no, if you think about our nervous system as electric, you know, it's electricity, right? If you think about the neuron, the cell body is a transformer, and these wires are throughout. Now, how these, so if this is the spinal cord, and I'll talk more, we'll talk in detail. I'm just giving you an overview what we want to say. So we know about this system is working there. Now here, the neurons will be here. We want to find out how these different neurons are there formed. And we also want to find out that how these neurons, they decide to, that send their wires that I need to go to this finger, I need to go to leg muscle, I need to go to this muscle or this muscle, right? And that's why this is critical. We say that the nerve, they don't degenerate, or they don't regenerate if some damage occurs. We never have, for example, if you see the lizard, you break the tail, tail come out. But if you cut the leg, leg never come out. New leg is not formed. That is all because the new nervous system cannot be formed. So it is critical to find out how these axons, they go where they need to go when we are developing. So we'll talk more about that. We'll talk about now the development of different kind of neurons in the spinal cord and how those axons, they go to the places where they need to go. So any doubt here so far? This was straightforward, like a lot of things to remember. But so for lab, you were able to do the